Hopefully you're not feeling this way, but I didn't have anything specifically related to what we're doing with genetics, so I went through my little folder and that's what I came up with. But we are continuing with the idea of mutations. Hard radiation, X radiation, gamma radiation can break your DNA. And if it breaks your DNA and you don't repair it properly, then the DNA is different from what it was supposed to be, and it's now a mutation. So any exposure to radiation can do that, but we actually are very good at fixing our DNA. Usually you need an exposure that breaks your DNA in multiple places, and then you don't put it together right again. We'll talk about that. This is why we treat cancer with radiation, because cancer cells are dividing like crazy. And we hit them really hard with a beam of radiation if we can. Well, we're going to talk about cancer later. And what we're hoping to do is break the DNA in a bunch of places so that when the cells divide, they can't get the pieces to go where they're supposed to go. They haven't copied them right, they can't grab them right, and so the two cells are missing things that they need, or they have extra things that they shouldn't have extra things of, and hopefully it kills the cells. In people that don't have cancer, exposure to radiation can do that as well. You've got to get a pretty hefty dose though. Now there's other things that people wonder about for mutations. If I were to take a fluorescent bulb out, we've got all these high tension power lines that run across the counties. If I went out in the middle of the night under one of these things, even though I wasn't very close to the wires, I could stand on the ground and hold the bulb up. And in most places, it would light up at least a little because the wires have electromagnetic fields around them, and that'll charge up the gases in the fluorescent bulb. And people wonder about being near electrical sources that have these electromagnetic fields and whether that has an effect on us, mutations or other kinds of things. We're full of charged particles, we're full of ions. So it kind of stands to reason that being in the presence of a bunch of electromagnetic fields might have an effect. But nobody's really been able to link it to anything. Years ago, they did a pretty massive study of line workers, guys for the electric company, who go up and they work around, they go in and out of these powerful electromagnetic fields all the time. And they checked them for all sorts of different diseases, and they kind of matched them with similar people who don't do that, for age and overall health and other kinds of things. And then they went looking for details. And they actually found out the line workers were overall more healthy than the other people, probably because they're outside doing physical activity. But there was no like cancer they got more, or any kind of brain effects. There's nothing that really, disease-wise, showed up more in the electrical workers than it did in the controls, the normal people they compared them to. So it doesn't really look like electromagnetic fields do anything. And of course, for the last few years, the question has been a different type of radiation. How about the radiation emitted by cell phones? It's what they call the microwave type of radiation, which is a very broad swath of different types of radiation. So yeah, it's also the thing in your oven, but cell phones are not working on that frequency. It's the thing in a policeman's radar gun. And they found out that years ago, when they used to have the handheld radar guns, that if they didn't turn them off and they just set them in their laps, that the radiation could cause testicular cancer in the cops. So there are certain frequencies that will cook a burger or cause cancer. And the question was, how about cell phones? Is that they don't emit or receive a tremendous amount of radiation, but they are very close to us. We're definitely within the zone. And the studies are all over the map. Is that some studies have found effects, a lot of studies have found no effects. They did a study on rats where they had rats raised with cell phone emitters right by where they were, and compared them to rats that were raised exactly the same way without cell phone emitters. And they did find that in the rats, there were physical differences in the brains when they got to be adults. But when they ran behavioral tests, they like rats because rats do a lot of behaviors that you can check. When they did behavioral checks, they couldn't really they couldn't link the changes in the brain to any kind of change in behavior. So who knows? Right now, the jury is definitely still out on cell phones. And there isn't any reason why the 5G is gonna be any more of an issue than the 4G. Yes, it's a little bit, it's flowing information a little bit better, but that's more of an efficiency thing rather than a radiation thing. At least I'm pretty sure that's true from what I've read. Here's something that we didn't even learn about when I learned about this back when I was in school. Your chromosomes, I can dig through here. Your chromosomes have little caps on them. It's a way to keep your repair system from thinking that these are broken ends and need to be stuck together, because that's what happens when your chromosomes break. You've got chemicals that go, whoa, broken ends, and they grab and put them together. And that happens all the time, but it's usually like a single break, and it's pretty easy to find the two ends and stick them together. If you get multiple breaks, that's where it's a problem, because there's no, oh, where did this come from kind of response. It's just, here's a broken end, but here's another broken end, doink. And sometimes you lose pieces that way, or stick the wrong things together. But the telomeres are like, almost like the ends of a shoelace. They're these caps. And okay, here's how you make a copy of your DNA. Imagine two long ropes next to each other, right? Your DNA's the molecule has two strands that you pull apart, and you make a copy on this side and a copy on that side to copy what used to be there. So when you do this, you clamp this huge enzyme onto your DNA, and it kind of rides along the DNA, and it makes a new copy on one of the strands. It rides along, the strands will come apart, it's zipping along one of the strands. It gets this big thing, gets down to the end, and it doesn't just zip off the end and keep making copies of everything that was there. It gets to the end and lets go. And so you've got a section that it used to be on that isn't two strands, it's only one strand. And you have another enzyme that has to come in and fix that. Make sure that you got the whole end of the chromosome made, the whole end of the telomere. It's called telomerase. And it's a little bit weird. Telomerase, there we go. If you have damage, you need to make more cells. You gotta make copies of your DNA to make more cells. Well, in a bunch of cells in your body, as you get older, your cells stop making this stuff. And so every time you make new copies of DNA to make new cells, the ends get more and more frayed. They get single instead of, because the next time you make it, you hit the end of where the old copy used to be and come off, and now it's more frayed than it was. And one of the things that will happen in a cell is, okay, we need more cells. The cell will go, well, let me check my DNA to see if it's okay. And if it's damaged in any particular way, including your telomeres are too frayed, your cell goes, I'm not making more copies. Have fun with the damage. We're not fixing the damage if you're looking for me to do it. So we accumulate and don't fix as much damage as we get older because of, for some reason, we've deactivated the telomerase gene in some cells. There's a bunch of cells where we don't. We're always making skin cells and lining cells through our life, and we're always making telomerase there. But in your deeper tissues where you do need to make repairs sometimes, it's a problem. And you do accumulate these messed up cells that also can be a problem. So this is very associated with aging. And so there's a bunch of people. Aging's a lot of money in studying aging because there's a lot of old people that don't want to age badly. So there's a lot of research, a lot of money into looking at telomerase. Here's the flip side. In order for cancers to really get rolling, they need to make lots and lots and lots of cells, and they would like to degrade as little as they can do while they're making lots and lots of cells. So your typical successful cancer, which is always kind of randomly activating and deactivating genes as it goes, a lot of mutations. Most cancers will activate the telomerase gene. So its chromosomes don't fray as it's making bunches and bunches of cells. Now, not too surprisingly, a lot of money in cancer as well. And so the cancer people are looking to shut this off. The aging people are looking to reactivate it. And the cancer people say to the aging people, well, if you reactivate it, you're going to give cancers a leg up because they won't need to come up with that random mutation. You'll have already given it to them. And the aging people are saying, well, if you're going to treat, you're going to go after this, you're going to produce aging effects in your patients. So maybe this is not something you want to go after if you're going after cancer. And right now, there's been studies. It turns out it's a difficult thing to target. And so far, the studies haven't really come down on one side or the other as to whether they really, one approach messes up the other one. But imagine cancer money, aging money. There's a lot of people looking at this stuff. And you'll hear about telomeres. They sell stuff on TV that's supposed to restore your telomeres. It doesn't, but that's what they say. And then there's epigenetics. Now I talked about my scalps dividing and a whole chromosome went extra into one cell, and you go, well, isn't that really, really bad to get an extra chromosome? Well, the thing is, by the time you're making scalp cells, those cells are not using every gene in your closet. They're just using the genes that they need in order to make the scalp work and produce hair follicles and that sort of thing. So it could be that hair color gene was the only significant gene that was in use in those cells, because the other ones were useful in other cells in other parts of your body. So different cells have to activate and deactivate certain genes in order to do what they need to do. So extra and missing chromosomes, once a cell is pretty well established and it's only using some of the genes, it can get by on that, hopefully. I mean, it gave me a patch of different hair. But we do that all the time. We're always activating and deactivating certain genes. And sometimes the timing gets messed up. Now, I'm lactose tolerant. Does that mean I have a mutation? Not really. When we are babies, lactose is the sugar that is packaged into milk. So it's a sugar, but it's different from sugars you get any other way except from dairy. And we all produce an enzyme to break that apart so we can absorb it and use it, because it's two sugars stuck together. Now, most mammals that nurse, they don't need to make that enzyme their whole lives. When they stop taking in milk, it's pointless to keep making it, so they deactivate it. Now, my ancestors from Europe raised and domesticated dairy animals. And so they had dairy as a potential source of food if they could digest the milk. There was other things in there that are useful, and you could do things with dairy that kind of minimizes the lactose. But there was a real evolutionary advantage to being able to access those sugars through your whole lifetime. So our timing changed over generation after generation after generation. And people that are lactose tolerant tend to come from populations as ancestors that raised domesticated dairy sources. Whether it was horse milk in East Asia, or elephant milk in parts of India, or goat's milk, I think, I'm not sure about goat's milk, or cow's milk, is that if you came from a population of ancestors where that didn't happen, then you likely turn off the lactase gene, and then lactose goes in one end of you and out the other, taking a decent amount of sugar with it, or a decent amount of water with it. And it can, mess with your tummy a bit. But It's an evolutionary trait, but it isn't completely a mutation, because epigenetics are more the processors are changing, and the timing of the processors are changing. And you can't make that happen. There's some people selling books that activate certain genes or deactivate. Yeah, you can't really do that with your mind. Okay, for the last part of the genetics thing, genetic technologies. We've developed a bunch of technologies that, from what we know about genes. One of them is we can analyze DNA. Forensics at a crime scene is that if you look at your handout about that, you don't analyze all of the genes or anything. You break the DNA up into particular bits that you can make migrate in a field and then stain them, and you get bars. And matching up, I think 14 bars, the more related somebody is, the more the bars line up. And there's a certain critical number of bars that tells you, whether you got a match in a sample. It's also what they do for most of the ancestry tests. If you go up and, your ancestors came from here, or they came from there. The question is whether they've got a really good read on these different populations from all over the world to really match you up, which is why you'll see articles about identical twins sending in and getting different results from the same supposed test for their ancestry, because there's a lot of statistical probabilities and iffy matching that goes on with genetic technologies, with that kind of what they call mapping. They also do screens that way, where if you have a genetic, an allele that's bad that runs in your family, you could potentially have, through amniocentesis, a genetic screening for that particular allele in a fetus to decide whether you want to keep the fetus or not. So there's mapping and screening and ancestry tests, and there's engineering. There's messing with it. GMOs, I was just talking at supper last night with my family about GMOs, genetically modified organisms. And there's a range of different things. Is that there's nothing particularly evil about GMOs as an approach. The most commonly farmed rice in Asia, at some point years ago, somebody took the gene to produce beta-carotene, to produce vitamin A, that rice does not naturally have. And they put it in rice and got it to stay there, and now the rice that's cultivated is actually vitamin A enriched because they put the gene for it in there. And that's what happens, is that you're taking genes from one place and putting them someplace else, most of the time for genetic engineering. And the question is, what exactly are you moving? And also, where is it being expressed in the particular organism that you put it into? Is if you're moving a frost resistance gene into, say, oranges, is it really being expressed in the oranges, or is it just being expressed in the leaves? Because we don't eat the leaves. And if it's being expressed, what does that mean? Well, it's producing a protein. You've taken the protein from another place that maybe people don't eat and put it into this crop. Is it showing up in the stuff that people are eating? And if it is, what could it possibly be doing to them over time? It makes the crops pest resistant, per se, like the corn borer resistant in corn. Well, that's being expressed in the corn cobs. So it's a really good chance that it's making that protein that's in the corn. And they got to test the stuff to see if it's safe. But the one thing they can't do is they can't test it for 20 years to see what the long-term effects are. And another thing about GMOs, genetically modified organisms, is a lot of the companies that do this have a reputation for being kind of evil. And so the process of genetic modification has been connected to this kind of, in it for the money and doing terrible things to farmers and other kinds of things, companies. and it's kind of stained the whole process. So it's one of those things where, sometimes it's good, sometimes it's bad. A lot of times it's a who knows kind of deal with GMOs. We do that also with, I mentioned that we used to get insulin from cows and pigs. Now we get human insulin. Now it's not being extracted from a human being. What we did was we isolated the insulin gene, and we got it transferred into things we could raise in the lab, bacteria to start with, and I think they're using mostly yeast now. And so you have these big vats of these single-celled organisms that are cranking out human insulin, which people would develop allergies to cow and pig insulin because it's slightly different enough that some people's immune systems recognized it. It works okay, but some people's immune systems went, oh, this isn't ours, and would produce antibodies to it. They don't do that to the human insulin that's made. And we do that with human growth hormone, which we'll talk about later, and a whole bunch of products that are made by human genes that we can actually get these organisms in the lab to make for us. And then we can have human versions of those. Related to this are stem cells, where we try to genetically, or these replacement cells that we have in our bodies, the ones that the telomerase goes away with, and they don't want to make replacements. But if you can take these replacement cells out of an embryo and put them in where their cells are damaged, they will make replacements for the damaged cells. If you take them from an embryo that's not you put them in, say your brain for Parkinson's, your immune system kind of checks them and doesn't find anything foreign on them because they're embryo cells. They don't really have much on them. But once they go into your damaged part of your brains and make brain cells, they are now the brain cells of somebody that's not you. And one of the problems they wind up with these stem cell transfers is that your immune system will eventually flag them as foreign. So now they're trying to take a cell out of you and mess with it in the lab to convince it to be like an embryonic cell. So they can put it back in you, and your immune system will see it as part of you. It gets very complicated. Cloning is kind of related to that. Okay, we've got a very brief thing coming up that I'll cover.